In this video, we will talk a bit about genes and brains as they relate to dyslexia. The thing to begin with is the understanding that every mental or psychological function or process is performed by the brain. There's nothing else to perform it. So this means that every mental, psychological function or process is a biological function and process. And this means that differences in performance that are observed between individuals are attributed to difference in the cognitive processing mechanisms of these individuals, which are attributed in differences in the brains of these individuals. So this is a distribution of performance showing different individuals along an axis of performance, better performance on this end, poor performance on this end. Most people are around average, and these people differ in their performance because their cognitive processes are not equally effective or equally efficient, and that's because their brains are different to the extent that matches the differences in their outcomes. So these differences are traceable to brain differences. How do genes get into this picture? Well, brains develop partly on the basis of the genetic instructions. So genes are one contributing factor to brain development. They're one strongly constraining factor to brain development. So at least some part of these differences is due to genetic differences, and that part is called heritability. These blobs here, as you almost certainly know, are chromosomes. So genes are found on chromosomes. The human cells have 23 pairs of chromosomes in their nuclei, and each chromosome is composed by two strands of DNA, and these two strands are complementary and code for the same genes in two versions. Each person has, for each chromosome, one version from their mother, one version from their parent, and what they pass on to their children is some combination from the two versions that they have. So if these two are the two DNA strands on one chromosome for the mother, then these two are blended randomly to create two blended versions, randomly mixing the components from the mother's mother and the mother's father. And for the father, the same is true. So this is inherited by the father's mother, and this is by the father's father. And these two are mixed by randomly chopping and combining so that these two people will pass on a random combination to their child. One child of these two parents will receive this combination. Another child will receive another random combination. What about twins? There are two kinds of twins. Identical twins receive exactly the same random combination of DNA from their grandmothers and grandfathers because they split off after conception. So monozygotic twins, identical twins, share 100% of their genes. In contrast, dizygotic twins are like regular siblings, except they're simultaneously conceived, and so they have different combinations of their parents, that is their grandparents' DNA. On average, they share 50% of their genes. And it is this critical difference along with the fact that monozygotic and dizygotic twins are born and raised at the same time in the same environment that allow us to estimate the effects of genes in the differences between various skills or diagnostic categories. So two twins that are born and raised in the same family, the same environment, are assumed to share 100% of their environment. They're assumed to grow up exactly in the same environment and to be subject to the exact same environmental influences, at least the common ones. There is also an error term for the non-shared environmental influences, whatever may apply to each one of them. So that's accounted for. These are the common environmental influences. And then there is the part that is accounted for by their genes. So if what we're measuring is affected by their genes, then if the two twins are identical, it would be equally affected by the genes. So these will be correlated 1. If they're non-identical twins, then they would be affected by genes, but not equally, because these are only correlated at 50%. So they share 50% of their genes. So we can use the different kinds of twins by measuring the extent to which their performance or their diagnosis are correlated in order to quantify the genetic effects on this skill or diagnosis. In other words, to estimate heritability, first of all, we note that identical twins share 100% of their genes. Non-identical twins share 50% of their genes on average. Both kinds of twins presumably share environment approximately to the same degree, and the comparison between the two kinds of twins exposes the effect of the genetic difference. Let's see how that might work out in an example for a diagnostic classification. Let's say this concerns dyslexia. So we recruit a group of children who are members of twin pairs and are diagnosed with dyslexia. So our probands are individuals with dyslexia who have a twin. And our probands are going to come from the low end of the distribution of reading skill. And this is the arbitrary cutoff that defines whether or not they're called dyslexic. And the average performance of our probands, because they have dyslexia, they come from this region of performance range. The performance of our probands on average is these gray bars here. So they're always here because these are our probands. Now, what do we expect to see in their twins? So remember, each proband who has diagnosis of dyslexia also has a twin. If dyslexia is completely determined by genetics, this is a hypothesis, it's not. It's a theoretical hypothesis. If dyslexia would be completely determined by genes, then for monozygotic twins, if our proband has dyslexia, then the twin also must have dyslexia because they have exactly the same genes. So we would expect the twin to fall here as well. For the dizygotic twins, we do not expect the twin to have this performance because they only share half of their genes. So if dyslexia is completely determined by genes, we expect the twin to be somewhere nearer the average, but not at the average. We expect the twin to have an increased probability of having dyslexia, but not the same as the proband. So these are the predictions if dyslexia was completely determined by genes. If dyslexia is completely due to systematic shared environment. If this was just an environmental issue, genes had nothing to do with it, and it was perfectly predictable by the environment. What would our predictions be? Well, both monozygotic and dizygotic twins, so both identical and non-identical twins, share their environment to the same extent, and they share it completely. So for both monozygotic and dizygotic twins, we would expect the twin of the proband, so the twin of the person with dyslexia, to also have dyslexia all the time, both for identical and for non-identical twins. So these are the predictions for pure environmental cause, and these are different from the predictions for pure genetic cause. And if dyslexia was random, it was neither due to genes at all, and neither to the environment at all, just caused by random circumstances, then we would expect the twins of the probands to have average reading performance because they wouldn't be affected by the common genes or by the common environment. We would expect them to have an equal probability of having dyslexia as anybody else. So these are the predictions for just error variance, no shared environment effects, no genetic effects. These three hypothetical predictions are the basis for understanding what we do with the actual findings. So if we analyze actual scores from actual twin pairs, what we will see is that compared to the probands, the monozygotic, the identical twins, are closer to the probands than the non-identical twins, which are closer than average. So there is some effect of environment and some effect of genetics. And by comparing the distance between the proband and the monozygotic twin on average, we estimate the non-shared environment. So this is the random, the unpredictable effect. Because monozygotic twins share both genes and environment. So any distance here is our non-shared component. And then that is true also, the non-shared component we assume is the same for everyone. So that would put the non-identical twin here, but the non-identical twin is actually there. What is this difference? Well, this is the difference that is caused by the fact that they share only half of their genes. So this difference is the half of the genetic effect. So by observing the actual performance of identical and non-identical twins of diagnosed individuals, we can estimate the components of genetic, shared, and non-shared environment and the extent to which they contribute to differences in performance on the relevant skill, here reading, that is relevant for the dyslexia diagnosis. So that's how this works in conception. These kinds of findings must be interpreted with great caution. First of all, the estimates are not independent. The estimates for genetic, shared, and non-shared environment are constrained by the way these hypotheses work and by the fact that they must add up to 1. So if something increases in a given study, something else must decrease. All effects are proportions of variability. They aren't actually estimating the level of reading skill or any other level. They concern differences between individuals. That's what the proportions can account for. And they're affected by any existing sources of variability. So if environments are uniform among the twin pairs, so if all twin pairs are raised in a very similar kind of environment, that will inflate the estimate of heritability because there will not be a lot of variability in the environment to account for variability in performance. And conversely, if we sample our twin pairs from very diverse environments, then the heritability estimate will go down because there will be more variability in environment to account for variability in differences in outcomes. Moreover, heritability estimates are not a measure of biological determinism. They cannot be that. First, because we're not estimating the level of the outcome, and second, because they're so specific to the assumptions and to the environment in which a particular sample was sampled. They're just indices of variance components for this specific sample. And so you should never conclude from any study of this sort that instruction or intervention doesn't matter. It's still possible that it may not matter for some things, but these kinds of studies cannot lead you to conclude that because they cannot answer that question. Having clarified that, we can say that heritability indices, so alpha squared as they're usually known in the literature, is in the range of 0.4 to 0.8 for more things. Actually, in one study, passage comprehension heritability for fourth graders was estimated to a staggering 0.86. But these are otherwise quite commonly seen numbers, and in general, the estimated heritability for reading-related performance, so these are test scores, or for diagnosis, these are classifications, but the estimated heritability of both differences in performance and classifications are in the range of 50 to 80% very commonly. So this is a very substantial proportion of variance that is accounted for by genetic factors. It is also substantially lower than 1. That means that there is a lot of variability between individuals that is not accounted for by genetic factors. What is it accounted for? Well, genetics cannot account for everything because genes don't function in isolation. They cannot predetermine anything. Genes are just portions of DNA, and they can constrain development. They can co-determine it by being expressed in a particular environment at a particular point in time in the context of their history. So whatever has happened up to that point. It makes sense to ask about genetic effects, like which genes are involved, which genes are affected if we have a diagnosis, what is the functional outcome of differences in genes for brain development, what is the functional outcome in terms of neuropsychological processes, what are the educational implications of that. These are all reasonable questions that one may ask concerning genetic effects. Let us turn a little bit our attention to brain function. A lot of researchers for decades have been working on mapping different brain areas, delineating different areas in the human brain and how they relate to specific kinds of functions. And this is one proposed map for language-related functions, including expressive and receptive language, including auditory and visual. In other words, spoken and written language. There are other efforts to map elements of computational models on the brain to locate the hypothesized functional elements of models. This is the triangle model of reading, and there are efforts to locate these components on specific brain areas that are assumed to perform that function. And there are different mappings depending on what people measure and how they measure it. But there is increasing convergence. There is also a lot of effort by researchers concerned with how reading is accomplished by the brain, who are trying to find the signature of dyslexia in the brain. So how does a dyslexic brain look like? There are a lot of studies comparing the brains of groups of people with dyslexia to the brains of groups of people without dyslexia. Unfortunately, our understanding of the reading network in the brain is nowhere near sufficiently developed to be able to say very much about that. So we should be very careful in our interpretation of the findings regarding dyslexia. First of all, there are many inconsistent findings. Some of the findings have turned out to be consistent. Many aren't. There are more people working on this, so we expect that eventually there will be more consistent findings. However, the fact that groups differ in some aspect of brain function does not imply that one of the two groups is abnormal. Researchers have frequently documented differences in some brain function between groups of people who are differentially skilled in some domain. This does not imply that one set of brains is abnormal, because researchers then do not try to study the individual brains of people in one group, for example, dyslexic group, and see if they fall outside the normal range on anything. So at the moment, there is no evidence for abnormality in the brain of people with dyslexia at all. There are several findings concerning structure and other findings concerning function. Structure is anatomy and function is activation. And the two sets of findings only converge to a limited degree. We expect more research that will lead to a more consistent and better converging set of findings, because any differences in function must be supported by differences in structure. And finally, a very important question that has not been resolved to any significant extent is the question of causal direction. If we compare a group of people with reading difficulty and a group of people without reading difficulty on a reading task, and then we find that there are differences in their brain function or even in their brain structure, this doesn't mean that this difference in the brain is causing the difference in reading performance. It is quite possible and even likely that the differences in the brain are the outcome, are the result of the differences in learning to read. Long-time reading experience of differential effectiveness will lead to differential development of the brain, because the brain continuously develops and rewires and adjusts itself in response to the challenges of its usage and the outcome of its usage. In other words, the brain is continuously remodeled and continuously learning based on what it does. And any differences in brains between two people who have had differential effectiveness in learning something is quite likely the result of that difference rather than its cause. Of course, it is even more likely that differences in brains and differences in performance are interacting such that there is a reciprocal causation, so that initial brain differences lead to differential effectiveness in learning, which lead to increased differences in brains or other differences in brains, which contribute further to differential effectiveness in learning, so that there are diverging trajectories leading to very different performances and very different brains. In short, the findings of researchers on the brains of people with dyslexia are at the moment entirely inconclusive. Two things to keep in mind are that there are brain regions that are involved in reading processes. There is nothing surprising about that because how else could we read? It's brain processing that does the reading. And the function of the reading regions varies among individuals, which is again extremely unsurprising because how else could reading performance differ? The only way for reading performance to differ is if the brains function differentially. So all we have at the moment is anatomical, functional, and behavioral heterogeneity. People differ in reading performance, and some are called dyslexic. And that is because people's cognitive processes differ in effectiveness and efficiency. And that's because people's brains differ in structure and function. And that's in part because people have different genes, and in part because people are subject to different environmental effects during their development. So this is variability among individuals at all levels, and an arbitrary cutoff here which selects the lowest performing individuals and labeling them dyslexic. According to this view, and consistent with all available evidence, this view of dyslexia is not correct. This view says that during development, normal development leads to typically reading children, whereas some cause of dyslexia disrupts development and leads to a child with dyslexia. This is the neurodevelopmental view. There is no evidence for anything like that. Instead, the evidence we have is that there is normal variability at all levels during development, such that some children are exceptionally good readers, others are average, lower than average, and some are sufficiently lower than average that are labeled dyslexic due to an arbitrary cutoff that we set, but there is no qualitative difference in here except normal variability in developmental trajectories across levels and domains of description.